Hello, welcome back to the third session of Success in Neuroscience Symposium. This session is on advancements in Alzheimer's and dementia. And we have three speakers who, again, will follow the same structure as we've had before, 15 minutes followed by questions. Now, Alzheimer's and dementia are not glamorous diseases. There is no cure or miraculous recovery. However, there is still amazing work underway on how to better detect the diseases, prevent them in our populations, and support those who are diagnosed to live a happy and fulfilling life. This session will look not only at genetics and causes, but at caretakers' roles and all the fantastic work that's going on in between. So, our first presenter is Dr. Satyabrata Kar. Dr. Kar's laboratory works on two complementary research projects. The first project is designed to address the underlying cause of neurodegeneration in the brain of Alzheimer's disease patients. The second project is aimed to study the role of insulin-like growth factors, IGFs, in the normal brain and their implications in Alzheimer's disease pathology. And the overall objective of these two projects is to establish the cause of the preferential vulnerability of neurons in Alzheimer's disease and how to determine how these neurons can be protected. Dr. Kar, great to have you with us. I turn the session over to you.
Thank you, Alan, and thank you for the Campus Alberta Neuroscience for inviting me. And we can start the slides. The title of my presentations, as you can see from the slides, a nanoparticle-based treatment strategy for Alzheimer's disease pathology. This is a little bit of a novel project, and I am a basic researcher. I'm looking at the cell levels, animal levels, but little bit of on cells derived from the Alzheimer patients. So what I will do, for the sake of convenience, I'll give a brief introductory notes about Alzheimer's disease and how one molecule is called beta-amyloid peptides, their aggregations and accumulations in the brain can trigger degenerations of neurons and subsequent development of the disease pathology. Then I will show you some of the recent data where we found that a family of small molecules called PLGA nanoparticles, which have already been approved for delivery of drugs in humans, they can able to inhibit the aggregations of the beta-amyloid peptide in test tube. And also we can show you how these molecules can able to prevent the toxicity in cultured neurons grown in Petri dish. Then we'll show you how these molecules can able to inhibit or at least reduce the pathology associated in animal models of Alzheimer's disease. It is 5-XFAD mouse. Then also show you some data how it can also prevent the toxicity of the neurons derived from the Alzheimer's patients. So now coming to the introductions, can I have the next slide, please? Alzheimer's disease, as you all know, is a progressive neurodegenerative disorder characterized by gradual loss of memory, followed by deteriorations of higher cognitive functions, which includes your language, praxis, judgments, and calculations. It affects approximately 6 to 8% of populations over the 65 years of age, but that's not important. Important is that the prevalence of the disease doubles every 5 years thereafter. If you calculate it, about 35% of the populations over 85 years of age, they do suffer from Alzheimer-related disorders. To visualize the impact of the disease on an individual, what I did, I have selected the self-portrait of an American-born artist named Mr. William Uttermohans, as you can see on the right-hand side, who was diagnosed with Alzheimer's disease at the age of 61 in 1995. This self-portrait, as you can see, provided a remarkable insight about the impact of the disease on the brain and mind of an individual in a short span of 5 years. That is 96 to the 2000s. You can see how the self-portrait is changed from normal to an abstract. And given the fact that in this current world, we have approximately 50 million people living with Alzheimer's disease, and with a steady increase in our life expectancy, the number of these people suffering from the disease will increase from 50 to 152 million people by 2050 unless we develop a better treatment strategy than the current one. By the way, I will tell you, this disease was first identified by a German psychiatrist named Dr. Alois Alzheimer in 1906. And after 100 years, it is found to be the most prevalent cause of dementia affecting elderly people in our society. If you look at the etiology of the disease, only minority of cases, less than 10% cases, the disease develops due to the mutations of specific genes. So far, 3 genes have been identified whose mutations can lead to the development of the disease. That is, the genes are amyloid precursors protein, presenilin 1 and presenilin 2 genes. Whereas the majority of the cases, more than 90% cases, are sporadic. There is no mutations or any type of direct associations, mutations of the genes can lead to the development of the disease. However, certain factors have been shown to increase the risk of developing Alzheimer's disease. One is the age, female gender, stress, lower educations, and most importantly, high midlife cholesterol levels. If you look at the pathology of the disease, as is shown in the figures in the bottom, you do expect to see a gross brain atrophy. On the right-hand side, it shows the eddy brains. There is a drastic atrophy compared to the normal brain. And also you see the loss of neurons in selected brain regions. Apart from them, you expect to see some extracellular neuritic plaques, which is shown the brown colored deposits identify the red arrows on the right-hand sides, which contains a peptide called the beta amyloid peptides. And also you expect to see some intracellular neurofibrillary tangles marked with a violet arrows on the right-hand pictures, which are present within the neurons containing another type of proteins called tau proteins. Unfortunately, at present, there is no cure for Alzheimer's disease. Can I have the next slide, please? The drugs that are available for the treatments, that is donepezils, rivastigmines, galantamines, memantines, they provide only symptomatic relief for a fractions of AD patients for a certain period of time. The recently approved A-beta antibodies, the aducanumab and lecanemab, their profiles or the efficacy, long-term efficacy still remains to be established. It has only been approved last year and this year. So now the question is how the disease is developed. Extensive research over the last 30 years have shown that prolonged increase in the levels of the beta amyloid peptide in the brains can lead to their accumulations, which can subsequently lead to the loss of neuron, cognitive deficits, and development of the disease pathology. Given the significance of these molecules, majority of the drug development strategies are targeted towards reductions or regulating these productions of the levels of the A-beta peptide or beta amyloid peptides. That means how to reduce the A-beta productions, how to inhibit its aggregations, how to increase its clearance, or how to inhibit the toxicity induced by the peptide, as shown in the figures on the bottom sides. So there are different targets are there identified with the arrows plus the stars. And this is the major targets of drug development. However, a major limiting factors in developing effective treatment strategy for Alzheimer patients, can I have the next slides, please, is the presence of a blood-brain barrier, which prevents the entry of any potential drugs or agents to the brain. Over the last decades, various nanoparticles are very small engineered materials measured about less than 100 nanometer in length, have been studied extensively, and they are used to deliver the drugs to specific regions of the body, including the brain. And in case of Alzheimer's disease, nanoparticle used to deliver the drug, at least in the experimental conditions, to the brain, where they can regulate A-beta productions, aggregations, clearance, toxicity. Indeed, a family of nanoparticles called PLGA nanoparticles, which are biodegradable and biocompatible and clinically used in delivery drug in a human, when those drugs are conjugated with some agents like donepezils, curcumins, and others, they can have beneficial effects in attenuating Alzheimer's disease pathology in cellular animal models of Alzheimer's disease. However, very little is currently known how the PLJ nanoparticles themselves, without conjugations with specific molecules, can have an effect in Alzheimer's disease pathology. Can I have the next slide, please? That's what we did. We accidentally found that this nanoparticle can have an effect on Alzheimer's disease pathology. And here I will show you some evidence. We studied the effects of this nanoparticle without conjugations of any drugs on the aggregations of A beta peptide measured in test tubes, and measured an effect of the nanoparticles on beta amyloid toxicity measured in neurons grown in petri dish. An effect of nanoparticles in animal models of Alzheimer's disease, this 5X FAD mice, which exhibits cognitive deficit as well as beta amyloid containing neuritic plaques in their brain. Finally, I'll show you one data where you can able to see the effects of these nanoparticles on neurons derived from Alzheimer patients. Can I have the next slide, please? This shows the aggregations of the A beta peptides measured in the test tube in presence of a dye called thioflavulin T. As you can see from the graph, the beta amyloid peptides aggregates with increasing concentrations from black, pink to green, and that reaches 6 hours, then becomes stabilizes. And this is validated by fluorescence microscopic studies shown in the green, as well as on the electron microscopic studies shown in the brown color, in light colored pictures below the fluorescence imaging study, where you say that at 0 hour, beta amyloid peptide don't aggregate. But at 4 hours, there is some aggregations, but 24 hours, they become aggregate and starts forming fibrils. This is validated by another assay called dynamic light scattering, or DLS, where you can see beta amyloid peptide at 0 hour, they are measuring size about 10 to 100 nanometers. Where at 24 hours, this same beta amyloid peptides, they aggregate and form bigger size, which measures about 1000 to 10,000 nanometer in diameter. Subsequently, what we did, we aggregated the beta amyloid peptides in the presence and absence of PLGA nanoparticles. Can I have the next slide, please? As you can see, the PLGA nanoparticles dose-dependently inhibited aggregations of beta amyloid peptides. And this is shown in the TSTSS and also in the fluorescence imaging, where you can see much less number of aggregated beta amyloid peptide present in presence of PLJ than in the absence of PLJ, which is shown in the green. Then also in the electron microscopic studies and native page gel electrophoresis, where you can see in presence of PLGA, the amount of aggregation goes down. So this shows that PLGA can inhibit the aggregations of A beta peptide. To determine whether PLGA nanoparticles can trigger the disassembly of already aggregated A beta peptide, which are usually found in the Alzheimer patients, what we did, we aggregated the A beta peptide, then exposed to the different concentrations of PLGA nanoparticles. Can I have the next slide, please? As you can see here, the PLGA nanoparticle dose-dependently triggered the disassembly of already aggregated beta amyloid peptides. This is shown in the kinetic assay, as well as in the fluorescence microscopy, electron microscopy, and gel electrophoresis. So this data indicate that this nanoparticle, without conjugations with the drug, can inhibit the spontaneous aggregations of A beta and also can trigger disassembly of aggregated A beta peptides. Now coming to the significance of PLGA on beta amyloid toxicity, what we did, we performed a series of experiments on neurons grown in petri dish. Can I have the next slide, please? We and others have previously shown that the neurons grown in petri dish, when exposed to the beta amyloid peptide for a long period of time, they do die. And here on the left panel, you can see the beta amyloid treatments decrease the viability of neurons, and it simultaneously increases the number of dead cells, and which are also indicated on the pictures below. We see red label structure, those are the dead neurons. And the toxicities or the death of the neurons is mediated by some intracellular molecules or activations of these molecules called tau kinases, tau phosphorylations, productions of toxic free radicals. Those are shown in the Western blots, and also productions of increased levels of CLIP caspase 3. Next slide, please. However, when the same cultures were treated with the PLGA nanoparticles, they are found to be significantly protected against beta amyloid mediated toxicity. As you can see from the graphs on the top panels, the PLGA treated cell cultures are protected against the beta amyloid mediated toxicity, indicating that more cells are surviving when they are treated with PLJ. And that is being mediated by reducing the signaling, activations of the signaling molecules that are found to be present in beta amyloid mediated toxicity, including tau kinases, tau phosphorylations, and oxyradicals. As a follow-up to this, we wanted to determine if PLJ can able to reduce AD-related pathology in 5x FAD mouse models of Alzheimer's disease. Next one, please. These mice, they exhibit cognitive deficit as well as the beta amyloid containing neurotic plaques in the brain as observed in the human AD patients. To examine this, what we did, we administered PLJ nanoparticle continuously into the mouse brains for 28 days using Alsat mini osmotic pump. Then we evaluated the cognitive performance using object recognition memory, then other pathology. Object recognition memory, based on tendency of any normal in animal to spend more time exploring a novel object compared to an familiar object. As you can see from the graph on the top panel, the mice which are 5XFAD mice not treated with the PLGA, they can't able to discriminate between novel and familiar object, that they show exhibit cognitive deficit. Interestingly, the mice which are treated with the PLJ, they seem to spend more time exploring the novel object compared to the familiar object. That means indicating the restitutions of impaired object recognition memory. Simultaneously, we show that the levels of the increased beta amyloid peptide decreased in these mice following PLJ treatments. Next slide, please. In parallel, we show the beta amyloid containing neurotic plaques, shown in the pictures on the bottom panel, are drastically decreased in presence of the PLJ nanoparticle. Their number, size, area, it goes down. So next slide, please. After that, we establish how this PLJ nanoparticle can act on the neurons derived from the Alzheimer patients. We expose the neurons derived from the controls and Alzheimer patients to beta amyloid peptides, which are also found to be toxic, but PLJ is found to protect them against the toxicity as shown in the graph below. And upper panel shows the neurons from the controls and Alzheimer patients. Next one, please. The protective effect, as observed in the mouse, is found to be mediated by inhibiting or attenuating the signaling molecules which are associated with beta amyloid toxicity, like tau kinases, tau phosphorylations, and CLIP-caspase-3. So in summary, the results obtained so far, can I have the next, please? The PLJ nanoparticle without conjugations with any drugs can able to inhibit A-beta aggregations and able to trigger disassembly of matured A-beta aggregates. They can protect the cells against beta amyloid mediated toxicity, and these cells and the mouse derived from mouse brains. Chronic administrations of this nanoparticle can reverse cognitive deficit and also attenuate beta amyloid pathology in 5x FAD mouse models of Alzheimer's disease. And these nanoparticles are also found to protect neurons derived from Alzheimer's patients against beta amyloid mediated toxicity. Collectively, these results suggest that the native PLJ nanoparticles, which are clinically approved for the delivery of drug in human, may have some unique therapeutic potential in the treatments of Alzheimer's disease. Next slide, please. So these are the peoples, and this work is supported by CIHR, NSERC, and Campus Alberta Neuroscience, APRI Asant, and SINAT grants. Thank you.
Thank you. That's an extraordinary journey. Covered a lot of territory in a remarkably compressed time. Thank you very much for guiding us through that. A couple of questions have popped up in the window. Are there any adverse effects of using nanoparticles?
Okay, that's a fantastic question. These nanoparticles, if you look at the literature, we found it accidentally. It has been in clinic since 1970s. It is used to make, it is a biopolymer and biodegradable. And when these nanoparticle breakdowns, they form the normal cell molecules, which are taken up by the cells without any. But yes, it does have toxic effect if somebody uses very high concentration for a long period of time. In our studies, when the animals were treated for 28 days with the effective concentrations of the nanoparticle, we did not see any single toxic effects. Not a single animal died. None of them, they lost the body weight, did not show any type of impairments in terms of the behavior or other things. So depending on the dose, how it is used, if it is within the safety regions, yes, it can have an effect without having any toxicity. And now these nanoparticles, but just to let you know that currently used in delivering 15 drugs in humans, as it is used mostly drug delivery.
Okay. One more question before we move on. How feasible are nanoparticles on a large scale for treatment? Is it a scalable product?
Yes, I am not aware of being used for any neurodegenerative diseases, to be honest with you. Yes, it has been used in delivering drugs in cancer patients, but the uniqueness of our studies is that most of the people use this type of nanoparticle to carry the drug molecule to the target areas. But they did not look at whether this nanoparticle themselves has a therapeutic effect or not. That's what we did find, that the nanoparticle which can carry the drug, but they themselves can have a therapeutic effect. So this is a little bit of a novel areas. We don't know, to be honest with you.
Okay. Well, thank you very much for that remarkable tour. We'll move on to speaker number 2 in this session, which reminder again is titled Advancements in Alzheimer's and Dementia. And our next presenter is Dr. Athan Zovoilis. Dr. Zovoilis is the Canada Research Chair in RNA Bioinformatics and Genomics based at the University of Lethbridge. He is the director of the Southern Alberta Genome Sciences Centre and academic lead of the Alberta Bioinformatics Network. His work focuses on dissecting the role of non-coding RNAs in cellular response to stress, developing epigenome and epitranscriptome sequencing, bioinformatics, and artificial intelligence pipelines for studying the non-coding genome. And with that, I will turn it over to Dr. Zovoilis. Thank you very much for joining us.
Thank you very much, Alan. And I would like to thank the organizers for the opportunity to discuss part of what we're doing here in Lethbridge in neurodegenerative diseases, but from a different aspect from what was presented until now. Could I have my slides, please? Next slide, please. So my lab is located within 2 institutes, the Southern Alberta Genome Science Centre and the Canadian Centre of Behavioral Neurosciences. And that shows that sometimes in order to tackle some questions, we have to tackle them from different angles and through different disciplines. And that's what we're doing in Lethbridge. Could I please have the next one? And the previous speaker made an excellent presentation regarding basics of AD and the fact that we are expecting a tsunami of new cases. So more than ever, it is important to understand which are the mechanisms that underlie the pathogenesis of dementia, and particularly Alzheimer's disease, and identify potential targets. Could I have the next one, please? And indeed, during the last 50 years, we have an increasing number of publications that have tried to elucidate this, and the number of publications in AD continues to increase. Next slide, please. But now you can notice that there is a kind of discrepancy. So we do have a number of targets that have attracted special attention, and APP and its role in amyloid beta is on the top of the list. And then we have a long list of genes and pathways that have been implicated with AD, and we have significant work that has tried to elucidate their role and their connection with AD pathogenesis. And could I please have the next slide? As mentioned by the previous speaker, we still lack any drugs that really can cure AD. And of course, we also lack efficient diagnostic biomarkers to early identify the risk for AD and potentially intervene. And now the question is, why did we get to that point? Could I please have the next slide? So it is rather unlikely to believe that after so many billions of dollars invested in research of AD, there hasn't been really hard work devoted to this field. So maybe we have not been looking at the right place. And maybe we have not been able to do so because until today, we may have lacked appropriate tools. And could I please have the next slide? And that's something that we have to remember in case of AD. Late onset AD, as also mentioned by the previous speaker, is actually a multifactorial disease. Even if you have some of the high-risk genes, your chance to develop late onset AD increases just slightly. So at the end, we do come to the question, how it happens that for some of us, when we reach 60, 70, 80, we still maintain a very high level of cognitive impairment, while some others, already from their 60s, may develop Alzheimer's disease. This kind of sounds very destiny, and every time I'm thinking about this question, actually, I remember a Greek myth of the goddesses of destiny. So in that myth, actually, our life would be represented as a thread. And whenever one of the goddesses would decide to cut the thread, that would be the game over. Next slide, please. However, how well do we know the thread of life? Next. Until 20 years ago, most of research had focused on genes and part of the genome that encode for proteins. And all of the genes that I actually showed in the previous slide are genes that encode for specific proteins. However, those genes constitute less than 3% of the human genome. And the other 97% for a long time has been regarded in terms as junk DNA or noise produced during transcription of this part of the genome into RNAs. And it has taken significant amount of time, literally hundreds of publications, to start challenging this idea. Because people have always claimed that how can it be that you have 97% that's kind of junk? Then what's the meaning for the cell to spend so much energy to replicate this DNA, pass this to next generations, and so on, if this is useless? So what it turned out earlier during this century through the ENCODE project is that actually a big part of this known coding genome is transcribed into RNAs. And this made this debate even more hot because not only we have the DNA that's propagated across generations, but we actually spend energy to produce RNAs from this part of the genome. So if these RNAs are produced there, there must be for some reason. Next slide, please. However, if we go again through the publications in the field and what percentage of them have focused on this non-coding part of the genome, we get actually less than 1.2% from the overall number of publications. So for something that accounts for 97% of the genome that we carry, only 1.2% of the publication has been devoted into understanding its role in Alzheimer's disease. Next. Next. And why did this happen? It has not. It's not surprise if we consider that the amount of information, and I'm going to come to this again to this point in a couple of minutes, the amount of information that we have been looking for, this 97%, is really vast. And until 2005, the whole gene bank, which is a database that stores DNA from all organisms that have been tested until that time, was not more than 50 gigabases. In a sense, not more than around the DNA of 20 individuals sequenced today. So it's not surprise that people have regarded this part of the genome as junk, because practically we couldn't study it. And we couldn't study this because we didn't have the tools to read this information. So what really changed the last 20 years? And actually, that's a kind of a story by itself, because it shows how the changes in a new technology called next generation sequencing kind of brought together science fiction with reality. Next slide, please. And I mentioned this about science fiction and reality because some of you may have had the opportunity 20 years ago to watch that movie from Hollywood. Its name was Gattaca, and as we can see in the next slide, in that film, practically we have been able to read our genomic material in a matter of minutes. For example, here we can see the main hero of that film. A few minutes after he was born, the nurse took just a little blood from its toe, put this into a machine, and then start getting a list of diseases and the risk to develop those. Now, that was science fiction, okay? 20 years ago, we couldn't do it, but this is what we're getting today. Everyone can have their genome sequenced, and there is an increasing number of companies and consortia that can provide a yet not complete, but much better picture of our chances of developing a number of these diseases. So how did we get there? Next. We know that information that makes all our genome is in DNA. Next. And we can imagine this information as letters written on a book. So for example, one page with a font 12 would be one gene. But if we want to include this information for the whole genome, next, we would need actually more than 1,200 books, 1,000 pages each. Next. Now, until 20 years ago, to read just one page from this book, we would need more than a day. Today, next, we can read all 1,200 books with a sequencer that small that can fit in our hands. And in fact, and we can see in the next slide, we can do this simultaneously for a big number of people. So all the books for every person at the same time doing this for 48 or even more individuals. And this has transformed medicine and is going to transform also Alzheimer's disease. It is just that until now, there has not been many resources devoted in this kind of genomic research in Alzheimer's disease. But where research funds have been devoted, we could have very important results. Next. So we have precision health that many of you have heard. So genomics could play an important role. And a very good example, as we can see in the next slide, is actually what cancer scientists already have done. So the case of this patient here who had late stage cancer, and I will come very fast why this could be also very important. She was dying, not responding to chemotherapy. They sequenced her tumor, and then they found that genes that are high expressed there are found in people that have high blood pressure. Then they prescribe to her, to get together with her chemotherapy, high blood pressure pills, and the tumor vanished. And if you would ask an oncologist, they will tell that that's a miracle, and it is a miracle. So as we can go to the next slide, we can understand that as the cost per genome literally becomes close to zero, we have now for the first time the ability to search. We have the tools to search the same thing also in Alzheimer's disease. Next. So what we're doing here in Lethbridge is that we are applying a pipeline where we're getting samples from patients, post-mortem tissue, mouse model animals of AD. We applied sequencing, we analyze, and we try to find and associate changes in the genome, and especially the non-coding genome, with the disease phenotype, in our quest to identify whether there are other locations, in addition to the known ones I showed, that may be associated with AD. Next. So we have a pipeline, we are using computer sciences, and that's important because, as I mentioned, the amount of data is really immense. And we generate files that go through our pipeline and compare healthy and AD individuals. Next. And I'm going to close with just an example what this research has brought. So we have actually been able to identify some of these locations which are actually highly repetitive in our genome to be connected with Alzheimer's disease. Next. As mentioned by the previous speaker, next. Response to stress is connected with AD. Next. And we know that in aging and AD, our circle of responding to stress is abrogated. So we questioned and we sequenced and we used this new technology to check which genes actually are abrogated. Next. So what we did find is that while in the healthy hippocampus, and that's the primary target of AD, we do have, during response to stress, a moment where some genes go up and some genes go down, but after the stress, we have like a kind of peace in the brain, everything returns to the primary state. In case of Alzheimer's disease, we actually have continuous activation, and we can see this in the next slide, where while on the top, we can see that we have a pre-stress state, stimulus, and a post-stress state. In case of neurodegenerative diseases, and that's below, we actually have continuous stimuli that continuously lead to increase of these genes. Next. And we found the same also. The previous one was in mouse models of AD. We find exactly the same in human, and again, in these non-coding elements, RNA non-coding elements that control expression of these genes. And I'm going to close with my last slide, with the next slide, which actually shows that these changes in these genes highly correlate with the stage of AD of patients tested. So there is a significant diagnostic potential. This is what we're testing right now. And this is just one part of the non-coding genome we have tried to dissect. And with that, I'd like to go to the last slide and thanks everyone that has supported our research, people in my lab, the funding agencies, and of course, the invaluable collaborators. Thank you very much.
Thank you, Dr. Zovoilis. Again, so much information being delivered, and I guess to go from a sort of deeply quantitative analytical view to from the lay perspective I bring to the table, because I am not a neuroscientist. Where does the optimism sort of sit here? What is the next great barrier that you think will come down when you're looking at this sort of research?
Yeah, I think the next barrier will be actually understanding better this kind of language that we can read through the next generation sequencers. Because I think most of you have heard about the DNA, the 4 bases, the same we have in RNA, A, T, C, G, or A, U, C, G. But then we realized that there are chemical modifications, and instead of having only 4 letters, we actually have more than 120. So we just see the tip of the iceberg here regarding the level of information. But that's the good news, because as long as there are things that we don't know, this makes it more likely that what we have missed until now in AD research may lie there. So I think that's the next frontier.
Right. And it's nice to have some powerful computers around to manage more than 100 bases.
Oh yeah, that we definitely need that.
Okay, thank you very much. We're going to head to our final presenter on this session on Alzheimer's and dementia. Laura Vogelsang is a registered nurse and assistant professor at the University of Lethbridge. She presently serves as the president of the Canadian Association of Medical Surgical Nurses. Her teaching and research interests include digital health, healthy aging, and nursing education. She's also active on social media, and she gets today's award for the best Instagram handle, which I am going to follow as soon as this session is over, at Nurse It Like You Mean It. And with that, I will turn things over to Laura. Laura, the floor is yours.
Great. Thank you so much. And thank you for having me. This is a great opportunity. And before I'll start, I'll just share the news that the project I'm presenting, I'll actually be defending as part of my dissertation next month. So I'm a doctoral student on top of the rest of that bio, but hopefully you don't have to use the word student for much longer. So thank you again for having me. And if we can bring up the slides, I'll jump right in. Great. So my project is titled Using Immersive Virtual Reality to Build Self-Efficacy in Dementia Care. So this project maybe deviates a little bit. It's a bit more operational in the sense that I'll be focusing kind of on the care of dementia clients and caregivers' roles and how to hopefully build some self-confidence in that area, because lots of times I think we forget that attached to the disease is still a person, a loved one, a parent, a grandparent, and someone who's deeply cared about. And though they might not be the ones to benefit from some of the curative research, ultimately they still need care and dignity. So that's a little bit of the background of this project. If we want to jump to the next slide, please. So I won't belabor this point. I think my 2 colleagues who went ahead of me did a great job of introducing dementia and acknowledging that numbers are on the rise, both worldwide and in Canada. Certainly in our lifetime, we're going to see increases of the disease up to 166% within the next 30 years. So this work is important. More people are being diagnosed. People are living longer. And I had the privilege last fall of teaching a global health class to some of the nursing students. And we talked about high-income country diseases versus low-income country diseases. And certainly in places like Canada, where we're fortunate to have robust research and healthcare, we're able to help people live longer. But with living longer, that means we see higher incidences of these diseases in our country, and ultimately more people needing care. So if we want to go to the next slide, please. Like in my introduction, it was shown that I do have many roles. I do still work as a frontline registered nurse. I worked throughout the COVID-19 pandemic, both as a nurse and with students. Some of my role of being a nursing educator has been taking students into the clinical setting. Through the eyes of a healthcare worker, having practiced there and taken students there. By nature of the disease, sometimes individuals with dementia can behave aggressively. This is part of their disease process, not necessarily within their nature. It can lead to them having risk for injury. It can lead for risk to injury for the caregiver as well. So the healthcare aide, the nurse, etc. This can contribute to workplace absenteeism, higher costs for employers, and ultimately just more turnover. Within the student population, which as we know are our future workforce, I'm sure it's no secret to anyone attending this presentation right now that there is a lot of vacancies for registered nurses. The Canadian Association of Nursing Schools recently released that there's over 20,000 vacant positions for nurses across Canada. And I can tell you, we are not graduating 20,000 nurses per year. So unfortunately, I think this problem is getting worse. To put it bluntly, working in long-term care settings is not sexy. Many students don't want to go there. If students do begin their careers there, oftentimes they move on. So there's just challenges with continuity of care and really having people who want to, work and thrive and have their careers there. It's certainly an area I've enjoyed working myself. So clients are more at risk for injury. It can lead to lower staffing ratios. If there's, sick calls or people away who are injured, we just don't have the manpower to care for these individuals and give them the care that they need. Another challenge is that time and funds for professional development are low. So this just hasn't been a priority to allocate money into for this ongoing training that healthcare professionals need to ultimately provide the safe, effective, and dignified care that we know all of these individuals deserve. One of the challenges is oftentimes with formal professional development programs that maybe is a course or a seminar I would attend on a work day. So now I've left a vacancy that then another nurse would have to fill, and we just don't have that. So out of this, if we want to head to the next slide, please, came the Caregiver Project. I was fortunate to team up with the VR folks down here in Lethbridge, and we were funded to explore different health solutions into ultimately keeping healthcare workers working in this area and providing better care for our clients. So within this project, we were funded to create 3 virtual avatars and scenarios that someone can work through, ultimately with the goal of improving your self-efficacy or your self-confidence to manage the aggressive behaviors that clients with dementia may exhibit. So sort of with the rationale that if you feel more confident, literature has shown that you ultimately produce a better outcome and you act more in an evidence-based way. So we do know that when people don't feel confident, they perhaps don't intervene as quickly. They're unable to de-escalate without physical restraints or pharmaceutical interventions. So giving, antipsychotics, sedatives, etc., which a client may otherwise not need. So when I talk about this project, I often allude to the books many of us read as children, those choose your own adventure novels where, Timmy fell down the well. Do you go get help or do you go down after him? Turn to page 64. It's very similar to that the individual can put on the goggles or the VR headset and work through a different scenario, choosing different options and ultimately hoping to de-escalate the aggressive behaviors of the client without needing chemical or physical intervention. We built some neat features into the VR platform. We recognize that there's no black and white algorithm for managing dementia, and what works well on one day you might do the exact same thing the next day and have a different outcome. So we did sort of build those things through the back end of our program to have different options or to have it reset. The scenario that I did my research on really was designed to actually make the caregiver frustrated and ultimately have to self-regulate their own emotions and not project that onto the client, as perhaps the client was refusing care or not willing to participate. So it's quite exciting. When we designed it through a collaboration with Red Iron Labs in Calgary, it is sort of now built with the infrastructure that we can add more scenarios. So the first scenario I designed was Vivian. She was somewhat the matriarch who managed care for everybody else in her life prior to her diagnosis of dementia, and now is becoming frustrated because she wants to maintain her independence, to get up and get ready for the day, but cognitively and strength-wise just isn't able to do so. So if we want to go to the next slide, please. Essentially, my little piece of the pie was looking at nursing students because that's who I know and work with. But essentially, I did want to see if there was any significant changes before and after running the VR simulation with the student group, and then what their perceptions were of how it affected their self-efficacy. And then if some of those self-efficacy traits were maintained after they actually went to the clinical setting in long-term care to care for this population. Next slide, please. So ultimately, within the study and my Likert scale, the inventory of geriatric nursing self-efficacy, which was a pre-existing scale I had permission to use, we did find that there was a significant increase in the individual students that did use the virtual reality platform, did feel more confident to then go into the dementia setting and care for this population. And like I said before, there is literature to suggest that if someone goes into an area feeling more confident, ultimately they enjoy working there more, they provide better, safer care, and it's hopefully somewhere that they will want to stay and work and enjoy and thrive in their career. So the control group versus the intervention group, it was higher. And the students, after leaving the clinical setting, also had higher self-efficacy scores than those who did not following practicum. So essentially, the results did show that this platform could be used to improve self-efficacy within students. I haven't had the opportunity to work with registered professionals yet. Next slide, please. I also was able to hold qualitative focus group interviews after the students were done their clinical rotations. And some of the main themes that came out of asking students about their experience using the virtual reality and how it related to their clinical long-term care experience. And just for context, they did 144 hours in a long-term care in Southern Alberta. For the themes that were identified, benefits of VR, working with this population, they felt like it provided them real world experience. So when we look at virtual reality and we're designing different, scenarios, two things people look at is presence and immersion. So essentially that realism, we put a lot of detail into designing the room. There's drawings from the grandkids up on the wall. The brass light has a little bit of reflection on it, really to bring that 3D element, and then that ability to interact with different things in the room. Again, starting in the beta, there's different objects. You can help Vivian wash her face, brush her hair, and hopefully as we build into this, there'll just be more and more ways that the individual using virtual reality can interact. Students felt that it created that safe place to practice. So oftentimes students do feel scared to make mistakes, that they're being evaluated. And they did feel that this, they knew they weren't going to hurt a client. They knew they weren't going to get hurt themselves. Interestingly, watching the students as a third person, Vivian at times would escalate and reach out to perhaps strike the student, but they knew they couldn't actually get hurt unless you walk into the wall, which I did once. So that was an accident. They did feel that it helped them to build empathy for the human experience of living with dementia. And they recognized that this was a way to help meet clients where they're at and just sort of come in with that appreciative approach of being present in the moment, not necessarily, seeking to cure a disease that can't be cured, but just ultimately providing a good day for the client and doing what you can in that moment. One finding, though, was that students did feel that this was an adjunct tool to their education. It couldn't replace that real clinical experience that one does have going into the setting, but they did feel that it was really great for the repetitive nature. So the ability to do this over and over again, possible remediation. If perhaps a student really is struggling in the clinical setting, they could go use this to practice and hopefully build those skills in order to move forward in their education and ultimately their career. I'll just invite the next slide, please. So some implications for moving forward. Of course, we don't want to just leave it with doing this small study with students. We're designing 3 scenarios and hopefully more, depending on where the research goes. I would love to evaluate it with formal registered health professionals, so registered nurses, healthcare aides, licensed practical nurses. One really great benefit of this is VR compared to simulation is relatively cheap, which is always a good thing. So a headset would be, it would be feasible for a business to own a headset or two for under $1,000 compared to some of our simulation mannequins that can run upwards of $50,000. So I think there is, to an extent, some cost effectiveness within this. And then that idea of bringing the education to the setting versus having the individual have to leave for possibly seminars and having that less immersive, more passive learning. Sorry, I think someone has a question.
Oh,
All right.
We'll get the questions at the end.
Sorry, I'll just keep going. I thought someone unmuted. I apologize. So in terms of that administrative lens, certainly opportunities to hopefully keep workers in the workplace thriving and wanting to do this work and stay in this setting, and then making it an appealing place for students because they feel, more confident, less scared that they're going to get hurt or injured. So yeah, I think it's a growing area of research, again, recognizing that it's possibly remediation and not a way to totally replace or revolutionize the way we're educating students. We still value those important clinical hours. Further opportunities, certainly, for evaluation research as well. And you can see in the video there, that's Vivian. I speak about her like she's a real person, but you can see some of the detail I alluded to within the virtual reality environment, the pictures on the wall, etc. So I think this is an exciting emerging area. Virtual reality is something that certainly nursing researchers are paying more attention to in terms of meeting our educational outcomes in a way that's cost effective and maybe has that long-term sustainability. So I apologize, I tend to talk very quickly, especially with something that I get excited about. But I think for me, just being a registered nurse that's worked in these places, ultimately we have all been touched by Alzheimer's or dementia in one way or another, and just seeing that opportunity to improve the outcome for the person who's really living with it right now, hopefully, rather than have to move towards chemical or physical intervention that could potentially cause hurt or cause harm. So I'll invite the next slide. And this is my final thank you slide just to Alberta Innovates for the opportunity to be part of this project. And then I just want to mention some of the collaborators I work with. I was sort of brought in as someone that has the perspective of how to develop a dementia scenario, but Red Iron Labs in Calgary, as well as the virtual reality folks at Lethbridge College that helped to bring the vision to life and ultimately make it a virtual reality experience for our healthcare workers. So thank you.
Thank you, Laura. That was fascinating. That really and compelling. I think there's a good question here for you, and I have a second part to the question as well. I want to add from my own personal experience. The question is, although the focus is on training the upcoming nursing classes, has there been any thought of using this platform with current registered nurses to help further educate the current workforce and provide them with that additional confidence?
Absolutely, and that is the direction that we hope to go, especially as it's perhaps brought to market for those individuals. So our population, just to sort of see if there was any effect of using it, with students, to be honest, a lot to do with their accessibility within the nursing program. But absolutely, we see this as being valuable for healthcare aides, nurses, practical nurses, as well as ongoing professional development.
And what about that next great leap? Because many of us have sort of frontline experience with elderly parents. I watched my father spend 6 or 7 years looking after my mother as she degenerated with Alzheimer's, and kept her at home as long as he could. And of course, you care for her, but the caregiver needs so much attention. And I think about a tool like this helping my elderly father, just because he's got no tools. He's flying by the seat of his pants, and it was so hard for him. And these sorts of things, as technology becomes more ubiquitous, maybe there's even a way to, or do you think it's a reasonable or responsible way to move it into the more public sector?
I do, and I think there has been increased interest. I use that term formal caregiver versus informal, meaning someone, perhaps a family member. I think there's huge opportunity there, especially because family members, they know their loved ones baseline, and they're perhaps quite scared to see those behavior changes and really not sure how to navigate or manage that. So that's definitely a next step I would love to explore more. And I know from some community associations down here in Southern Alberta, they're interested as well, even just for perhaps business owners and members of the community, how they can recognize and support an individual if they perhaps were witnessing some areas of memory loss or whatever. So absolutely, I think perhaps even with an augmented reality lens as well, there's opportunity there just to kind of see the individual in their home, even look at, how can we rearrange furniture better? How can we, get some of those supports in place? So, yeah. And that's another important thing to consider, I think, because the numbers are quite staggering. And is there going to be a bed for everyone that maybe needs care in that formal setting? Or do we need more capacity for informal caregivers to provide that care longer than perhaps they've had to in the past?
Another question, and then we'll start bringing the rest of the panel into the discussion. Is the virtual reality tool able to be used for more than just Alzheimer's care?
I think so. So when we kind of built in, again, that underlying structure that designs it kind of as the choose your own adventure, pick your, you say something to her, and then you have your option of how to respond. We did kind of build it with that baseline skeleton that I would love to move it into other areas. Or I think another area that we really need some professional development for nurses and healthcare workers is those who don't work in long-term care that maybe don't see this as often. So maybe I always say people with dementia still get sick and have other ailments. So once that person, gets so sick, they have to maybe go to the emergency room, and the emergency nurse is really good at handling the disease in the emergency. They're not so used to the layer of dementia complexity on top of that. This could be a way to support them to feel more confident to care for those clients in perhaps those other settings. So yeah, I think it could, we hope it's designed to be transferable as the different needs and trends in healthcare change.
Okay, well, let's invite Satyabrata and Athan back into the conversation. And while we're doing that, you can read there's a nice compliment there from Tammy about a timely and necessary tool you developed. Source of deep concern that our population is aging in such numbers while care diminishes. I appreciate your work in this area.
Thank you. That's very kind.
Yes.
Thank you.
Gentlemen, good to have you back again. I see pictures, I see questions popping up here. Svenja, when you say big picture, you see future opportunities in this field. Are you talking specifically to Laura or to Alzheimer's research in general? In general. Okay, so a question to all three of you, and we'll go in the order in which you presented. From Svenja, more of a big picture question. What do you see as future opportunities in this field of Alzheimer's and dementia research? What's needed to make these things happen? Are there opportunities that are not being realized, or that are nascent or embryonic, that are exciting? Dr. Kar?
Yes. I think one thing I must mention that if you look at the recent trend, and specifically in certain countries, the prevalence of dementia, especially in developed countries, is slightly started going down. But the absolute number is increasing because of we are living longer. The reasons for that, there was a nice review in Lancet. So reasons for that, the people are becoming aware of it, apart from the pharmaceutical therapeutic treatment strategies, aware of how the disease, the risk factors are there. They have started doing certain exercise, keep on learning all the times. Those are what is doing, it is delaying the development of the disease. That's how the number is going down in some countries, not all of the countries, and specifically in the developed countries, although the absolute number is increasing. In terms of the strategies of developing therapeutic treatments, yes, recently there are 2 beta monoclonal antibodies have been approved, although their efficacy needs to be established after long-term treatments. But there are a lot of drugs under currently development, and they may have a beneficial effects. So yes, it is a devastating disease. There is no doubt about that. And the number is going to go up. And we need to be prepared from all aspects. And drug development, it doesn't happen overnight. It takes 1 decade minimum to take one from lab to the human. And that is a little bit difficult to predict. When are you going to get an effective treatment strategy? But other ways we can able to mitigate to some extent, taking care of the patients, as Laura said, and also educating the people what they need to do. So keeping them active, keeping them learnings, and those things can able to help them to some extent.
All right. Thank you. Dr. Zovoilis.
Yeah, actually, I will echo what Dr. Kar said, and I would like to add here, we have to understand that Alzheimer's disease is a very heterogeneous disease to such an extent that it may not be actually accurate to speak for just one disease. It can be literally as many diseases as there are the patients out there. So applying a more personalized approach, I think what's the next to expect here. For example, it could be that some medications work with some a specific subset of patients, but doesn't work with another. I'm sorry, with another subset. And when we do the clinical trials, if the patients are not stratified correctly, we may lose that potential effect. So being able to stratify and being able to define a specific genetic profile for every one of them may help actually to find which specific treatments may be suitable for that specific individual. And until a few years ago, this was not feasible because of the cost of sequencing. But nowadays, it comes that it's cheaper to do sequencing than maybe go, for example, to a movie theater if you're not interested for a big number of sequences. So that's something that I think that we should expect to see within the next few years.
Okay. Laura, from a frontline worker perspective, where do you see potential?
Yeah, I think for me, the biggest thing is just actually getting the technology to the people who need it and will ultimately benefit from it. This technology is great, but it's not any use to anyone if it's sitting as an app on my desktop and not being utilized for frontline workers. So to me, it's just, I see my role now really in that sort of knowledge translation advocacy piece to continue to emphasize this important work, make it known about, the needs of my view and my priority is the needs of the nurses and the healthcare workers, and I certainly appreciate the body of work that's going on as well. But I just think it's really now moving into that, getting it marketed and to the people who need it, because one of the biggest benefits of virtual reality that we're seeing now is how portable and cost effective it is. And we often see that learning happens when people can be immersed and interactive versus me doing a PowerPoint presentation to masses about ways to de-escalate aggressive dementia clients. So I see that kind of as my next step, especially as this research kind of winds up.
Can I add one thing, Alan?
Yes.
Just to give you an advance, over the last 20 years, we have progressed quite a bit in terms of the diagnosis of the disease, imagings and other techniques, biomarkers. It is not perfect, I'm not telling it, but we have progressed quite a bit. In terms of the therapeutic, not except the two recent drugs, all are old. But the lifestyle factors seems to have a major roles when the disease can develop, at least in sporadic cases, not all cases. Familial cases are different, where the genetic mutations can lead to the development of the disease, that is different. Sporadic cases, the lifestyle factors may play a major role. Cholesterol levels and other factors, head trauma, stress, those play. And if those things are under our head and we can able to manage or control it, we may able to have a better control over the disease, at least non-therapeutic sides.
Right. Yeah, and the goal, of course, in any treatment is prevention. So
yes,
yeah. Well, in a similar vein, there's a question here for Drs. Kar and Zovoilis. How long after the onset of Alzheimer's disease are we able to use the tools you both proposed to start helping with some treatments? At what point? How long do you have before you begin the intervention?
So, Anton, you want to go, or Athan?
Yeah, I can go. So I think that the big game here is to do this before we develop the symptoms. Of course, for those people that have developed symptoms, we are looking to achieve a significant improvement. But given the nature of the disease, the earlier you get there, the better. And that's why the diagnosis before you get the symptoms is a very first important step. Identifying potential targets for these people before they develop symptoms and before you have increase in the levels of neurodegeneration is important. It is also important to consider that people that develop symptoms at a specific age, they develop symptoms at that specific age because there are no other cognitive reserves. But the process had started decades before, but until that time, the system was able to compensate. So the sooner we start, the better.
So, and one thing I must mention that, as Anthony said, that when it is being diagnosed as early as possible, then the treatment strategies can be used, but it is not practicable because the symptoms, because pathology started about 20 years before you start seeing this cognitive deficit, which is visible and clinically diagnosed.
Right.
So another interesting thing is that a lot of drugs we use for therapeutic treatments, which you use after the development of the disease, and still you can see that you can able to attenuate or reduce the progressions of the disease. And that happens quite a lot in animals. Somehow it does not get translated into human. All the animals models, you can see there are a lot of drugs are there. You can able to reverse means at least reduce pathology, but when it goes to the human, it fails. That is another major barrier. Certain drugs, I don't know how is it going to work. For example, I'm not telling that it's going to work for our type of drugs, which has been in the clinic since 1970s. Whether it will be useful or not, I don't know, because our results are very recent. But it seems to show that it can able to return or at least mitigate the disease pathology, both in the pathology as well as the behavior sides. And it is clinically approved and now being used without any problem. Whether it can be used in human or not, no idea. We have not gone that.
Okay. Rich fields to explore. Laura, a question specifically for you. We nurses in psych are seeing more Lewy body dementia. Am I reading that correctly? Are there examples in VR read this dementia mode?
Yeah, so to my knowledge, no, not yet. Even as I was doing my literature review for this project, this area is quite underexplored, especially with nurses as the focus population. So I guess the short answer is not yet, but I hope soon. Really, virtual reality is such an emerging technology for us that it's just getting off the ground. And this was kind of our first go, but I hope this continues to garner interest and that it will just continue to grow.
Okay. Well, we're right on time right now. I think this is a good way to end the third session. Thank you very much to the presenters. The subject matter is fascinating and so topical for all of us as our aging society confronts the reality of living so long. I wish you all the very best success as at the front lines of this research. So we are going to take a 15-minute break. We will be back for session number 4. We're going to the future. The future's over this way. The neuropreneurs will take the stage and talk about the business of neuroscience. So thanks again to our presenters, and we'll see you in 15 minutes at 2:30. Thanks, everybody.